Okay, well thank you everyone for coming. I'm really excited to be here. This is my first seagrass conference. And I'm sure you're all excited to hear some cool seagrass research, see some good photos from the field, maybe some dugongs and sea turtles. But I'm here today to get you excited about dirt. So, before you leave, just bear with me, because I think by the end of this talk, you'll be just as excited about using soil for seagrass conservation as I am. So when we try to restore seagrass systems, we try to get them back to their natural state. However, what is natural? How do we know what's been modified due to human impact? This relates to the shifting baseline syndrome, where each new generation perceives the natural environment as that which was first measured. So really we need long-term monitoring in order to observe what the system looked like before human impact. Long-term monitoring can also help us see what are the natural fluctuations in species composition and help us tailor management strategies that reflect a more informed desired state of the system. Essentially, we need long-term monitoring in order to manage seagrass communities in the Anthropocene. So my project is going to look at using DNA from sediment in order to reconstruct past seagrass community change. So other methods to monitor long-term change in seagrass communities include remote sensing work. This sort of approach is really good for obtaining information at a large spatial scale, but it's limited temporally, I think only about 70 years. Another method is using fossil data, so macrofossils, pollen. This has a much larger temporal resolution. However, it can be limited by being able to identify the remains. They can be degraded and sometimes not even being able to identify them. So my project's going to take sediment cores. I'm going to use stable isotopes in order to date the cores, and then I will extract DNA from the lengths of the core and use this to reconstruct coastal vegetation. So my project is focused on mangroves, seagrass and saltmarsh, but I'll try to keep it more to the seagrass side. So this is your typical coastline. You have seagrasses, mangrove, saltmarsh. And as time goes on, this system changes. So you can see that there's T1, T2, T3, and this is the changes through time. So if I was to take a sediment core here, I can see that I should be able to detect the change from saltmarsh to mangrove to seagrass. So hopefully that gives you a kind of visualisation of what I'm aiming to do. So this notion of taking sediment cores and extracting DNA in order to reconstruct past plant communities has actually been done before, and one study was actually able to observe how human agriculture impacted plant communities. Another study was actually able to extract DNA from sediment that was 400,000 years old. So as you can see, there's great potential for this sort of method. So these studies, in order to analyse the sediments, implemented a meta-barcoding approach. So traditionally when you want to identify a species, you'll extract the DNA, amplify a specific region, and then match the sequence to a database. Meta-barcoding means that you can take an environmental sample, extract the DNA, and you can actually, by amplifying one loci, you can identify many species in the sample. So I think the studies that previously have used soil to reconstruct plant communities used a tRNL barcode. So the approach that I want to take here is a hybridisation capture approach. So this can be done on just plant specimens by themselves or on environmental samples. And this approach involves using baits or RNA baits that bind the DNA of interest. And then you use a magnet to remove them, and you can wash away all your unwanted sequences. The beauty of this method is that I can target many loci to identify many species within the sample. So this brings me to a really important point, that this sort of method can only be used if you have an adequate reference database or reference library. That means getting specimens that have been properly morphologically identified and then generating sequences for these. So then when I take my environmental sample, I can be sure that what I'm identifying in that sample is definitely one of these species. So while this method is promising for seagrass conservation, there are still some assumptions and limitations to the method. So one of those is whether the DNA actually moves through the soil profile. So this is actually really quite hard to test. So I tried to do an experiment to test this, and I talked to soil hydrologists in order to interpret the results. And basically I can never replicate in the lab a true marine coastal sediment. There are forces and processes that go on that you can never truly replicate. So the assumption that other studies that have successfully used sediments to reconstruct plant communities, they take the assumption that when DNA is released from a cell, it binds to the soil and becomes part of the soil matrix. So essentially you want to know that when you take a soil sample and I'm saying that a certain seagrass species is there, I need to be sure that it was there at that time and hadn't moved through. So that's the assumption I'll be taking, that the DNA is bound to the soil profile. I can also actually check this when I do my soil cores. So I should be able to see a distinct change between seagrass, mangrove, saltmarsh. However, if I see a smear of the DNA signal, then it's more likely that DNA is leached. Another way that I'll be actually sort of confirming or validating my results is I'm doing some remote sensing analyses in the same area where I'm taking the cores. And although this will be a small portion of the core, it will still give me some external validation that my results are reflecting what's actually growing there at the time. So another sort of experiment that I did was to see whether this extracellular DNA, so the DNA outside of cells, or the DNA inside of cells, is what you extract when you extract from soil. And I did this by doing a normal soil kit, and most of the other studies actually just target extracellular DNA. They would wash the soil with what's known as a phosphate buffer, and it preferentially binds to the soil, releasing the extracellular DNA, and then you just analyse that. However, when I tried that method and another soil method, I found that I'm getting both intracellular DNA and extracellular. So this basically means that I am sure that when I'm doing my extractions, I'm getting the maximum amount of information that's there. So I want to be able to get all the DNA out so that I'm not missing species. So another limitation of analysing coastal or marine sediments is that there's many inhibitors. So things like humic substances, there's heavy metals, these can actually influence PCR processes. So one of the ways that I'm going to overcome this is the hybridisation capture approach that I mentioned before. So because I'm targeting my DNA of interest, the actual way in which I do that is also a 3D structure. So I'm actually pulling them out with magnets instead of what you normally have is just a filter. So I can actually get rid of a lot more of these inhibitors with this method. There's also more steps before the actual PCR, so there's more chance that I can get rid of the inhibitors. The other limitation for coastal sediments is that there's small amounts of DNA. So there's not a lot of DNA there, and the DNA is pretty degraded, especially in the ancient soils that I'll be analysing. So the hybridisation capture approach actually overcomes these as well, because it's targeting many loci, which means that it increases the probability that I'll actually be able to pick up the fragments of DNA that are there. So what I also did was I actually did some trials to see what the optimal extraction method would be for getting plant DNA out of soil. So your standard soil kit for DNA extraction actually targets microbial communities. So I did a series of tests to see what kind of kit would get me the maximum amount of plant DNA. So I trialled a soil kit, a plant kit, and I actually trialled a fecal kit, which sounds weird, but it had a inhibit-X buffer, which we were hoping would get rid of more inhibitors. And I also tried a magnetic bead kit, and this works by extracting the DNA through use of magnets, like the Hybe capture approach, so it gives that 3D sort of structure instead of a flat filter, so you should get rid of more inhibitors. Anyway, we found that the soil kit yielded the most DNA. But what I also did was, this kit actually contains glass beads, which could actually be broken by some of the shell material in soils. So we're actually replacing these with zirconium beads, and these are actually tougher and should actually lyse plant cells open easier. So basically I want to make sure that when I'm analysing these sediment samples, I need to be getting out as much plant material as possible. And I want to know exactly what's there. If I miss anything, then I'm not reconstructing the community properly. So what could this method actually mean for conservation in seagrasses? So we can actually use this to assess human impact, like I mentioned before, so we can see how humans have actually influenced the composition of seagrass communities. If I can take sediment cores that are deep enough and date these, I can observe some of the past responses to climate change events and see some of the resilience in seagrass species. This can help me and help everyone prioritise successful management strategies, so target those species that are more likely to survive into the future. And this can help us improve our conservation. So essentially this whole project is to use the past in order to inform the future and improve the way we can conserve and manage seagrass communities. So over the next, I think it's next month, I'm planning on starting my first trial with this method. So I guess all I can say is watch this space, and hopefully I can get out some data soon and we can look at the sort of applications that this method could possibly have. I just want to acknowledge some people. So the Goethe Institute funded some of my core sampling. Paul Lavery and Oscar Serrano kindly donated samples for me to use. I also want to thank the people who came up with this hybridisation capture method, so Ed, Core, Michelle. I want to thank them because without that my project wouldn't be so cool and innovative. Yeah, that's all. Thank you.
Okay, thanks Nicole, and we've got time for some questions.
Thank you for your talk. So you mentioned that you used remote sensing to see what's growing in your area. Could you please tell us what kind of data have you used?
Yeah, so we're using a model that was developed by DEW, which is Department of Environment and Water, in Adelaide. So they've actually used Landsat data and developed a model that reflects the whole state vegetation change. And so we're actually using their model and trying to just focus on mangrove and saltmarsh. At the moment we haven't actually got any results because we're trying to validate the model for that fine scale change. But yeah, that's what we're going to use. Yeah.
Okay, thank you.
Anyone else?
Really, it is an interesting ideology what you are proposing. And do you think the real-time diversity of seagrass will correlate with the result of the e-meta barcoding? What your result going to get?
Sorry, could you repeat that?
So real-time species, whatever diversity of the seagrass available in the particular ecosystem, when you are approaching through e-meta barcoding or otherwise hybridization, you are getting a result from the sediment based on the e-meta barcoding, you know.
I actually really don't understand the question, sorry.
Actually, you see, real time we are seeing the diversity of the species of seagrass in the particular ecosystem.
In real time?
Yeah. We are seeing the diversity of seagrass ecosystem real time.
Yeah.
But when you are approaching through e-meta barcoding of the sediments, whether you are sure the result will match with the real time data.
Yeah, well that's what I want to find out. So the whole thing about this method is it hasn't actually been done in coastal sediments, marine sediments. So I'm going to, trial the method and then find ways to make sure that it's actually reflecting what is going on. So one of the ways was the remote sensing. Another thing I could do is pair it with some fossil evidence that's been done. So yeah, I'll be making sure that I can actually validate before I definitively say, oh, this is the community structure. Yeah.
Okay, we are also seeing the same work in the mangroves of Andaman and Nicobar Islands. Presently we are concentrating on that.
Yeah.
Maybe future I will communicate with you.
Yeah, sounds good.